Quick announcement. We're doing, like usual, we're doing the sign-in sheet and the swiping card thing is right over here. So I'm really excited to get to introduce Dr. Nicole Barger. She is the assistant professor of ecology and evolutionary biology she's a assistant professor at associate.
The web page is updated.
Oh, she's got an outdated webpage. Sorry, I got my information from the wrong place. Okay, well, she's a professor at the University of Colorado, Boulder, where she studies arid lands ecology, and we're going to get to hear a little bit about that pretty soon. She started her science career at Evergreen State College in Washington State, and then moved on to the Department of Environmental Science Policy and Management at UC Berkeley for her master's, and then got her PhD at Colorado State University. So she's got a lot of diverse interests and pursuits within her research program as well as beyond it. Those include trying to improve STEM education in higher education, like we learned about last night. She's the chair of the science committee at Canyonlands Research Center. That's the beautiful picture up here. She is an active member of the growing community of 500 women scientists, and she's an author on a UN-supported land degradation initiative. So I could go on, but I'll give you some time to talk.
Thank you so much for that lovely introduction, Christine. And I just want to recognize Christine and Heidi for all the work they've done over the last 2 days hosting me. It's been really nice, and I really appreciate your time. And I've had another amazing day here. So many great conversations, so many wonderful graduate students, and all of you faculty should just be incredibly proud of these amazing graduate students you have. So with that, I am really happy to be talking about this biological soil crust restoration project. And I've been doing science for just over 20 years now, and this is the best project I've ever been on. It's the thing that I'm most excited about right now, and this is actually the first time I've had a chance to put all of this together. So what I have to do first, though, is start with the acknowledgments, because this is a massive project that you probably have friends up here. Sasha Reed, I know, is just here. So this is my dream team. I think I'm going to work with these people forever and ever, because they're just we all work really well together. There's amazing postdocs and graduate students in here, and my advice to any graduate students out there is that, like, once you find those people that you click with, and Kari and I were talking about that today, it's like, once you find them, never let them go. They're your friend forever, and like finding good collaborations are incredibly valuable. So here's my dream team. They contributed a huge amount to everything I'm going to show you today about this project. So what I want to do is start off with the really big picture. So what Christine said is that I am working on a UN-supported land degradation and restoration assessment globally right now. And so that sort of global perspective and looking at how lands are degraded, what are the potential for restoring some of these lands globally, really informs my work at this more regional scale back on the Colorado Plateau. So what we know, and these estimates are always really rough, is that an estimated 10 to 20% of dry lands are degraded globally, and these are primarily due to human activities such as livestock grazing, development, a lot of different types of agricultural activities. So this is just a very rough desertification vulnerability map. If we kind of zone in on this, where we are here in the US, you can see that, of course, we are in a highly vulnerable landscape for desertification. And many of you probably work on some of these issues of the impacts that humans have on these landscapes, on the plant communities, and on our soil resources. So a nice little happy note, following on some of those land degradation statistics, is that globally we are really starting to focus on how do we start restoring some of these ecosystems. So what we know from the area of the country that we live in now is that these landscapes have given us a lot over the last 150 to 200 years. We have had intensive oil and gas, more recently, development, livestock grazing, croplands. And so what we're thinking about now is how do we start restoring these landscapes? And what are some of the techniques that we can harness technology to start thinking about how to restore landscapes? So the nice thing is that from a global perspective, there is a real mandate coming down to start thinking about restoration. So last year, the UN Sustainable Development Goals, land degradation was put on the forefront of, and the idea of land degradation neutrality, which is when the area of productive land remains stable or increases. So if you want land degradation neutrality, and one area over here is becoming degraded, you need to be doing restoration in another area. So this conversation about restoration is becoming incredibly important. And restoration ecology has been a field for a while, but I think this global perspective is really going to start pushing it to the forefront. More recently, and in the near term, the bond challenge is that to restore 150 million hectares of deforested and degraded lands by 2020. I think they're nearly 80% to that goal. But what's interesting about this challenge is that it's really focused on forested lands and less so on dryland ecosystems. So that's what I'll be focusing on today, are drylands and the need for soil restoration in dryland ecosystems. So this type of aerial photo might be very familiar to many of you, as over the last 10 to 15 years, we are going through another increase in oil and gas development across the U.S. West. And so with that, you get soil surface disturbance or any other human activities may increase soil erosion, soil loss, alter nutrient cycles, and water cycles as we get these soil surface disturbances. And we also know, and many of us that have worked on Biocrust have told the story over and over, that recovery of Biocrust can be very slow. Like a lot of these landscapes, especially on the Colorado Plateau, for any of you that have tramped around these systems, you can see these beautiful Biocrust communities, especially in the national parks where there's less disturbance. So once you disturb these, the recovery can be fairly slow. And so what we've been thinking about is kind of harnessing our dream team technology, our expertise to come up with technologies and approaches to start restoring these soils. So for anyone that's just gotten here, like some of my Nigerian friends, that we were talking about soil crust, that let's just do a little Biocrust introduction really briefly. So biological soil crusts are functionally important in drylands because the big thing is they influence soil stability. So once you have a biological crust community there, in those vascular plant interspaces, they provide an incredible amount of stability and keep the soils in place. Once they're there, they influence infiltration and runoff and site hydrology, and also there's a lot of nitrogen fixers in the soil that influence nutrient availability, in addition to the photosynthetic capacity of some of these communities are quite high and can be equivalent to a neighboring vascular plant. So they develop on these soil surfaces. They're just in the top few millimeters, so there's all this incredible amount of microbial activity in those top few millimeters of soils. And the organisms that are largely there, and it depends on location and soil type, are communities of lichens, fungi, mosses, and cyanobacteria. So we know some of the long-term effects of soil disturbance by going back into landscapes that we know when it was disturbed, and then we can look at some of the recovery of those communities. So this is kind of a classic paper by Jane Belknap and Steve Warren, looking at tank tracks in the Mojave Desert from Operation Desert Strike. So these are Patton's tracks back from, and these data were collected 55 years after Operation Desert Strike in the Mojave Desert. So when they went back, and what you can see here, these are the old tracks. here in these light areas that were disturbed. These darker areas are the Biocrust communities that were undisturbed. So what they can do is go back and look at the cyanobacterial and lichen communities 55 years later. So what's interesting about this is that 55 years later, 46 to 65% of the cyanobacterial community has recovered, whereas only 3 to 6% of the lichen community has recovered. So when we start thinking about restoration, we know that we're not going to get the full suite of organisms back. And that these lichens are going to be more of a problem child than the cyanobacteria. But what we also do know is that when we start thinking about the function of these ecosystems, those cyanobacteria are those early successional cyanobacteria that come in early are really good at stabilizing soils and providing the function that we want to get back in the system. So we're less concerned about the species, getting all the species back in the restoration process than the ecosystem function. Okay, so the objectives in this project. So the big goal is to facilitate the recovery of degraded arid and semi-arid land soils by restoring these Biocrust communities. And the way that we're approaching this is by first establishing Biocrust nurseries. So just like horticulture, that we have nurseries of Biocrust that are these inoculum testing and supply centers for deployment. I like using deployment because this is funded by DoD. We put that in our title for fun. So deploying the inoculum into the field. But once we have developed the inoculum, we need to identify successful field methods for applying those. And what that means is that there have been failed experiments before where people grew inoculum, threw it out in the field, and all died. So we know that habitat likely needs to be altered in a way to promote restoration. And then once we have these developed, the third goal is to then evaluate all of these functional responses. So in terms of soil, how does it influence soil nutrient cycling, soil hydrology, soil stability, and we also threw into this proposal plant responses, but anyone that does plant restoration knows that that can be also incredibly difficult. So I won't be talking about those plant responses today. So just putting those goals into this kind of staged, multi-institutional approach, ASU is developing all the inoculum in Ferran Garcia-Pichel's lab. Northern Arizona and Matt Bowker's lab, they really did these very quick and fast field trials to say, how do we alter the environment to promote Biocrust growth in the field? And then once we made the inoculum, identified really good candidates for how we alter the habitat, we then started, and we're just 1 year into evaluating the kind of full multifactorial suite of different inoculum and candidates of how we manipulated the environment. So what I'm going to do is step through each one of these today. But first, what I want to do is present the framework in which we're thinking about restoration. And you know, a lot of this not just applies to biological soil crust, but really any restoration project. So along the scale, we have kind of the passive, you know, just what you need to do. This is increasing in the difficulty of the restoration project or the process. And so, I mean, obviously, the first thing you're going to have to do is remove any stressor from the environment. So whether or not it is for DoD training. So, I mean, the reason that this is funded by the Department of Defense is that we've had a lot of intensive training over the last 20 years in the Western U.S. military lands. This is foot traffic. These are tank tracks. And those have very different sort of impacts on the soil structure. So first is removal of the stressor, and then it's like, well, if you don't even have any propagules in the system for Biocrust growth, that's going to obviously be a limiting factor. So first is development of inoculum. Then as you increase in difficulty, then it's altering that environment in a way to either capture more water, You maybe have to water those biocrusts once they go to the field, altering that environment in which you're inoculating into. And then finally, if you have actively eroding, highly disturbed soils, you're going to have to artificially stabilize those soils before applying inoculum. So that's kind of the framework in which we approach this problem. Okay, so here are our two research sites. We chose a hot desert site in the Dryland Desert. So we are working at the Jornada experimental range in the hot desert. We were planning to work at Fort Bliss, but there's so much training going on right there that they couldn't even give us a hectare of land that they said they could protect. So what we did was we hopped over to the Jornada experimental range, which has very similar climate and soils, and that whatever restoration success we have there will likely apply to Fort Bliss. Here, this might be much more familiar to all of you. We're working at the Utah Test and Training Range, so it's about 45 minutes from Tooele and out in the Great Basin Desert. Okay, so first I want to talk about the development of the Biocrust inoculum and going back to the staged approach. So we have two types of inoculum that were developed at ASU. And so I'll go through each of these. So first, what we did is we collected, and this has always been a concern for everyone. It's like, well, where did you get these organisms? Everything was collected from these field sites. They're native to each of these field sites that we worked in. So we collected samples. In the first inoculum development, you can think about this as an easier, less microbiology intensive approach where small amounts of biocrust were collected from the field and then grown, and the biomass was increased in the greenhouse. So it's just like growing plants from like little starts. Then those were taken to the field experiments. So the second type of inoculum was lab-grown cultures, and those were from isolates of the different organisms, the early successional cyanobacteria in those field-collected samples that were then grown out into the field experiments. So from this general model, I'll go into more detail. So I want to first talk about this mixed isolate microbiology intensive approach. So this is our heroine, Anna Geraldo, a PhD student, and I have a little story about Anna in a couple minutes. But what she did is she collected these field samples, and she, using just microscopy, started picking out individual strands of the early successional cyanobacteria. So in green here, she's picking out the species that we want. We know that we want. And these W's are all the weeds that we don't want to culture into an inoculum. Then what she did is she scaled up the biomass, went through a hardening process, which is just similar to these isolates have been in the lab for a while, they're just not very hardy. They're not ready to be thrown out into the desert. So what she did is take them through this hardening process and then delivered them to the field experiments. So this is what she did first is when in collecting these field samples, it's asked the question of like, what's the composition first of the cyanobacterial community? So she actually did the full suite of cyanobacteria, bacterial, but then just to focus, and these are just the cyanobacteria species, what we found is here on the left two columns are the cold desert soils. These are the two hot desert soils. We're working on two soil texture types, so what I'll just be showing is silty and sandy soils. And so what you can see here, and what's been really interesting and shown over and over coming out of this lab, is these cold desert soils are dominated by Microcoleus vaginatus, whereas the hot desert, and this is a pretty new finding. We always thought that this was all the same species, but this is actually a hot desert species, Microcoleus steenstrupii. So this gives us a really good target of what are these cyanobacterial abundance in the soils, and what's our target for creating an inoculum, because we want to try to get as close to this as possible. So what she did then is she, like I said, isolated, cultured these, and then started doing a scaling up process. So this is a time scale in months. It was a pretty intensive process. It took about 3 months to isolate all of these strains and get them into a culture. And then she started growing them out and scaling from from small amounts up into half liter into these 20-liter vats. So as you can imagine, at some point, this probably has some commercial viability. We're discussing how we can leverage off of some of the biofuels efforts that are going on across the country, because it's a similar sort of culturing facilities in some ways. And so these are the main cyanobacterial species. So Microcoleus here. Nostoc and Cytonema are also important because there are nitrogen-fixing cyanobacteria. Okay, so once she had a sufficient amount of biomass, then it was like preparing the actual inoculum. So what she did is she took the biomass and hydrated it in with a growth medium, and a water mixture. Added that to a sterilized native soil. So this went back into a native soil and went first in the second to third day in these culture rooms with lights, so it's not a stressful environment. But then what she did is kept increasing the stress in this hardening process I mentioned. So from the culture room in days 4 to 8, it went into the greenhouse, then to the rooftop at ASU, increasing the stress of the environment with increasing incoming radiation. And then by day 15 or 16, they were completely dried, packed, and bagged and tagged for us. So during this time, what she did is she reduced the amount of shade. She went from 80% shade to 40% shade to no shade to increase the tolerance of these organisms to the increasing amount of sunlight. And she was also doing 11 wet and dry cycles, like wet it up, let them photosynthesize, but then dry them down. Because if you keep these organisms wet for too long, what you do is you promote all these other weedy contaminant species. So these wet-dry cycles are really important. Okay, so this, love Anna, she made tags with active ingredients, and there's 60% Microcoleus vaginatus in here. She flew in, so this is Anna's story, she flew in to the day before our field experiments started. She would not let anyone else take the inoculum to the University of Colorado from ASU. She carried them on the airplane, flew them in, delivered it to us. I took her out, of course, to a really nice lunch, and I said, "What are you doing tomorrow?" And she said, "I'm having surgery." I was like, "What? Why are you having surgery?" And she's like, "Well, I didn't tell you this, but 6 months ago, I was working and I broke a pipette in my hand. And it was bothering me after a couple weeks, so I went to the doctor, and the doctor said, you need to have surgery. And she said, I can't have surgery. I have to deliver inoculum. And so Anna worked for, I don't know how many months, 3 or 4 months, with a pipette in her hand, because she couldn't be off schedule to deliver the inoculum to us. And so she delivered this and then went and had surgery the next day. I mean, this is the sort of graduate students that we're working with, like, amazing. Anyway, that's Anna's story. I even have her x-rays somewhere in one of my talks. Okay, so that was one inoculum type that you're going to see. Here's a second one. Oh, I didn't mean that any graduate students needed to work like that. There was no pressure. So the second type, and then you can think of it as a little bit easier in some ways, that take small amounts of these field collected samples, take them into the greenhouse, we put them in just these small Tupperware containers on native soils, watered them. Well, actually, we didn't just water them. I'm going to get to the complicated part. So the idea was to enhance the biomass. However, the challenge is in these greenhouse environments is to keep that community stable and to not have it overtaken by a bunch of contaminants. So this is Sergio, and that is what he did as his postdoc, is he wanted to identify and manipulate the factors that constrain the growth of biocrusts. And so what we knew coming in is that there's a bunch of different things we could try. There's watering frequency, macronutrients, calcium, micronutrients, inoculum, like how you place the inoculum in there and how they grow in a slurry or in little mosaics, what's different levels of light intensity. So you can imagine if we were to do this in a full factorial, it would just overwhelm us. So what the approach that was interesting he took was this more of a screening approach, which is a fractional factorial. And so the idea with a fractional factorial is that you target specific treatments that will then inform, what are your best treatments. So it was a pretty unique statistical and experimental approach. So here's a lot of data that I will walk you through. So all of those experiments, from that fractional factorial, we had 18 different treatments that went forward. They're not marked because they're not necessarily important. I'll just give you the information in the next slide. But what I want to show you is that here we have the hot desert sites, sandy and silty, the cold desert sites, again, and the two texture types. And so these red lines here are the initial chlorophyll A content. So what we're reporting here is the chlorophyll A. This is just a nice indicator of the cyanobacterial biomass. It's just a nice biomass indicator. So what you see here on both of these, and so what the blue is the level of the chlorophyll A content of that initial, that inoculum that came from the field. So that's you can think about that as the target, is that's how much chlorophyll A content was in the field, and this is what came in initially once we only spread a little bit, because we only took a little bit of that and spread it like 10% cover onto those soils, so that's the initial amount. So what you see here is definitely across here some standout chlorophyll A content, some treatments that are obviously working pretty well. So just to simplify this a little bit, and it actually was a really nice, fairly simple story. And you can imagine with all those macronutrients and calcium that it could be a mess, but fortunately, what we found is that if we wanted to enhance the biomass and grow crusts in the greenhouse, we needed to decrease the light intensity, which meant shading with shade cloth. The water frequency was also at its highest, but again, there has to be drying cycles in between there, or the weedy species will take off. Same thing with the hot desert, but what's interesting about the hot desert was that in the silty soils, phosphorus was important, and in the sandy soils, nitrogen and phosphorus were an important nutrient to add to enhance the growth. Okay, so with that, going back to the development of this inoculum, the other question is that are we maintaining the composition of those communities? So again here, what I'm reporting are the cyanobacterial communities here. This is the hot desert sandy, hot desert silty, cold desert sandy, cold desert silty. The first bar is what came in and what the community looked like in the field. The second bar is what the community looked like at the end of growing out the cyanobacteria, or the biocrust in the greenhouse. And what we wanted to know is, like, have we altered the community composition significantly? And so as you can see, for the cold desert, these are pretty similar. So for example, these microcoleus, we do get a decrease in the abundance in microcoleus, but we have increased here, for example, the cytonema, which is an end fixer. So that's something we're like, well, I think that's actually it's still a pretty good composition because that's having those end fixers in there is important. So you can see that it's not perfect in terms of not altering the community composition too much from the initials, but we're still kind of working on now fine-tuning some of those wet-dry cycles, the water additions. So we felt pretty good here about not altering the community composition too much to go back out into the field as inoculum. So the other thing here is that making sure that we have enough biomass. So this, again, is at the end of all the experiments. These are the sandy and silty for both hot and cold desert. And what you can see is that here's the biomass, and we're far exceeding the initials here. So we have quite a bit of chlorophyll A content going back to the field. All right, so what I'm going to do is change gears now. While in those first 2 years of developing all that inoculum in the lab, what was going on at NAU is that they were conducting a huge number of field trials and since they didn't have inoculum from the lab yet, what they did was they just collected from nearby locations and used field-collected inoculum to conduct the field trials. So I'm not going to present all of the data today because it was 500 and some plots, but what they looked at was engineering the site with raking to roughen the soil surface. We also had water additions, shaded the soil surface to reduce incoming radiation. I was like hoping that wouldn't work because I didn't know how I was going to tell a land manager to shade the soil with shade cloth. It's just not reasonable, but we did it anyway. We also had, and what I want to focus on are the best candidates that actually went forward into here. Okay, so we checkmarked here, we developed the inoculum, and what we're talking about here is engineering the site in a way that will promote biocrust growth. So first experiment I'll present are what levels of inoculum do you need to add to get things going? And the idea here is that if you're going to harvest from this location and inoculate this location, you can't over-harvest because then you're just, it doesn't make any sense. But we're trying to get to the minimum amount of harvest from this location to then restore this location. And so what we decided here is that we would do 10%, 20%, and 40% cover. And so that just means for a very small amount of a meter squared over here would go onto another meter squared, only 10% in these experiments, or 20 or 40. So again, I was hoping that the 40 wouldn't work because I didn't want to over-harvest. And so the good news coming out of here is that, so this is just early ocular estimates of biocrust cover, so we don't have yet the chlorophyll A data. But what was interesting is that this low level of inoculum seemed to actually work pretty well, even on, so this is clay and sandy loam, this is actually the silty site, that this low level of inoculum was kind of good news because it seemed like, okay, we don't really need to go up to this high level of inoculum to get higher biocrust cover. So initially what was really surprising about this is that I was like, okay, we've told this story forever about how biocrusts take forever to respond, and this is a year later, and they're actually doing pretty well in this uninoculated disturbed soil control. So those hadn't gotten anything. What we did realize at this point, and this is how cool science is that our plots were in a sea. They weren't in a highly disturbed environment because these plots were in kind of a sea of biocrusts. And most likely what was happening is that there were propagules coming in, probably from overland flow or from dust, and we're probably getting a higher background than normally a large disturbed site would be. So we probably didn't have quite the propagule scarcity that we wanted in those initial trials, but that was okay. It still gave us, gave us some interesting data. So then we also looked at soil stabilization strategies. And we looked at two different types. One was the application of polyacrylamides. So we looked at three different types of polyacrylamides, and all this is glue. It's really compositionally similar to Elmer's glue. And that what this is used is to stabilize soil surfaces. It's a commercial product. These are three commercial products that are used to stabilize soil surfaces. So what we wanted to test here is that can we just apply this glue to the soil, put the inoculum on top, the inoculum will stick to it, and because we had concerns that if you have a highly mobile soil that's churning all the time and you throw really expensive made inoculum on it, it'll just blow away and disappear. So that was our first idea is to try these three different types of polyacrylamides. This straw checkerboard is it's a technique that comes out of China, and they have used, so this is just a square meter plot, but if you think about this, replicated across 20 kilometers to mobilize sand dunes in China, can kind of get that picture in your mind. That's what they're using in China to not have like these large sand dunes taking over roadways, railways, towns, because they have quite a bit of problem in the really arid parts of China. So they've developed this technique that we've also adopted here. So looking at those data, and what I'm showing here is just for the cold desert. We have, again, this is they called it clay loam here, but it's our silty site and our sandy site. The first bar in each of these, so there's control, PAM1, which is just the three types of polyacrylamides, PAM1, 2, and 3. And then the first bar is the control, and the second one is the inoculated. What was interesting here is that we're getting pretty high cover again in these controls. They haven't been inoculated. And after a year, we're at 50 to 60% cover of, and this is just, when we say Biocrust cover, this isn't the full suite of organisms. This is just the early successional and cyanobacteria that we call light crust. And these light crust means that you just can't see any of that, like really dark, and it doesn't have the topography that you would see down in some of the other like highly developed crusts like in the national parks. So what we saw here is like, well, there's not any PAM that's sticking out, as like really great. But then we looked at some of the late successional species, and there were some lichens that were popping up here. And just kind of averaging across, we decided, okay, it doesn't look like there's a clear winner in terms of the polyacrylamides, and based on some other stability data, we ended up choosing this product, which is actually called dirt glue, PAM2, to go forward into these big multifactorial experiments. So in terms of the straw checkerboard, here's some of the cover covers, just values from photos. So here we're looking at the control uninoculated Biocrust plots. So again, the straw checkerboard, all we did was went out to a ranch or a farm nearby in Twilla, bought a bunch of hay bales, you lay it on the soil surface and stick a shovel in, and that stands straight up. That's all. And I always imagined in China the amount of labor it took to do all these, a lot of mobilizing of sand dunes, but we don't have cheap labor like that in the US. Anyway, so these are the control plots. These are the inoculated plots. So what you can see, and these are all straw checkerboard, and as you can see, the inoculation here in the silty worked quite well. This sandy site wasn't as responsive, but also this inoculated sandy site also worked quite well. So what we did is we had those two types of inoculum that were developed, our two stabilization strategies that went forward now into what we're now really working on are these full restoration plots. Okay, so going forward, we now have in this final, we're in the final year of this project, we have this approach of 3 different types of inoculums. So we have our intact biocrust that are you can think about that as kind of a restoration target. No, we're not going to get there in terms of the composition, but we're looking at, how does that crust function in terms of stability and hydrology and nutrient cycling, and that's kind of our target crust. In implementing these experiments, we had to scrape and disturb and trample a bunch of plots. So what we did in scraping those plots is we saved all of that biocrust that we scraped and put in this inoculum treatment. So you can imagine this could be potentially pretty effective if you are an oil and gas company and you have some sort of mitigation strategy that you need to implement after you've gone in and put in a certain number of oil pads. Then you could scrape the surface, put that biocrust aside, and use that for restoration purposes later. So that's what that field collected, what we were really interested in. So this local biomass, there's Sergio again, that's the greenhouse grown. The mixed isolates is Anna, the lab-intensive development of the inoculum. So when you look at some of the cost benefits of these different inoculum types, the risk of over-harvesting is higher here in the field collected because you're collecting from the field, you're not growing it up in the greenhouse, and which decreases as you go toward this inoculum development. But these field collectors are going to be much better adapted to the environment. They never went into the greenhouse. They never had a nice, cushy life in the lab. So they're going to be more highly adapted to the local environment. And then the time and resource investment, I mean, this is really ultimately the users of this information are not only scientists, but really land managers. And so if they're interested in the time and resource investment, this field collected is the easiest way to restore an area. And your investment in time and resources increases from there. So the candidates that went forward in terms of soil stabilization were, again, the straw border and the polyacrylamide dirt glue. And there's my postdoc applying the dirt glue. And here's the full design. So again, we have our target. We had disturbed plots where no inoculum was applied, so just disturbed left open. Then disturbed with polyacrylamide only applied, no inoculum, and then just a straw border, no inoculum. So all of these plots were disturbed, and then we stepped through each of the field collected, local biomass, mixed isolates. Each were inoculated into either a polyacrylamide plot or a straw plot. There are a lot of plots out there. So these are both at the cold desert and hot desert site on 2 soil types, and what I'll present today are our very early results from the cold desert. So what I'm showing here are the chlorophyll A content of, this is the cold desert sites for both the silty and sandy soils. And so if we're going to have a target, our target level of chlorophyll A content of what's in the field in those intact crusts is around 30 to 50 micrograms per gram soil. So after 1 year, so 2015 is 3 weeks after the inoculation. We wanted to get an initial. So you can think about 2015 as kind of the initial, right after the experiments went in. 2016 was 1 year later. So I've highlighted these blues here just to try to separate all of these bars, because we're going to eventually present this better, but this is very preliminary right now. That right here is just the disturbed. This is disturbed, not inoculated, not with polyacrylamide and straw for each of the soil types. So you're already probably figuring out, it's like, wow, there's not like huge differences yet. So if we look at the 3 inoculum types, so in the pink is field collected, the light green is the local biomass from the greenhouse, and here's the mixed isolates. What we're So what I know in doing restoration, and anyone that does restoration, is that in the first year there can be a lot of noise. And this may be what we're seeing right now, is just a lot of noise in what is going on in terms of Biocrust growth after 1 year. But there is some suggestion that these field collected here and also here are actually doing pretty well. And we're not quite sure what's going to happen with the local biomass and the mixed isolates. And we actually had to have a secret conversation when Anna was coming to us and saying, "Where's your data? What's going on with my inoculum?" Because she'd worked so hard on this. And we're like, okay, we have to sit down and show Anna the data because what this is looking like, it's like, well, it's not really responding any differently than the non-inoculated plots right now. So we had to kind of have a gentle conversation about, well, it might not be responding yet, but it could next year. Anyway, so those are our early data, and we still have another year of monitoring this, and hopefully we're hoping to continue to try to find funding to monitor this over the time, because as many of you also know, with restoration work, some of the effects that we see are going to be possibly at year 4, 5, or 6, and not in years 1 and 2. So this is our continual challenge in doing restoration work, is that working in 3 to 5 year funding cycles, when your most interesting data may come in year 5 or 6 when your funding is gone. But the advice that I want to give on this is that we're learning to deal with this by partnering very early with the agencies that have these small amounts of money, because continual monitoring, oftentimes only costs about $10K to put a crew in the field and to get that work done. So we're getting around this kind of restoration issue by really going to our partners, BLM, DOD, Forest Service, whoever's land you're working on, and saying, listen, we just need to keep monitoring this over the long term to really understand how the system is responding to restoration. So here's our happy field crews out implementing the plots in both of our sites. And with that, I just want to acknowledge all the money our undergrads have gotten for doing undergraduate research out in the system, and also CERTIP for the support. This is a lot of support from DOD to do this project. And this is our happy dream team. And with that, I'll take questions. Yeah.
Kind of think about application in the future and BLM land, especially grazing. You do all this work, but if there's no changes in grazing patterns, the cows going to come back and destroy the areas that you're working to restore. Is there any plan for that?
Yeah, so that's a really interesting question. So for anyone that didn't hear, it's like, if you had, so with that framework, the first thing, you remove the stressor. So in that case, it would be livestock grazing. So clearly, we put in a proposal to ask that question, because most of the time in any restoration project on federal lands, it's a 2-year rest period before any animals are put back on. And so we've proposed to do a restoration project and then painfully put the animals back on after 2 years, after these restoration types, to actually show that this is probably an ineffective way. I mean, we actually probably know what will happen if you do a Biocrust restoration with plant community restoration and you put livestock back on after 2 years. It's probably not going to work, but it's just making a point with data. So we actually have that proposal in right now. But it is a big issue, is that if you're going to invest a lot of money into doing something like this, you're going to have to remove the stressor in a significant way. And I think you would only go on this track if you're willing to do that. Yeah. Yeah.
So when were the applications put out? Put in the fall or spring?
Yeah, so that we looked at the climate data, and we were trying to optimize precipitation because so working in the cold desert, we looked at the rain events and that we wanted the temperature to be just right. And so we know that for cyanobacterial growth, it's right around 20 degrees C. That is good, like 18 to 20, or even lower, actually, in the cold desert. But then we wanted to take advantage of the rains because they're going to need water to establish right away. So for the cold desert, we put them in May. And the hot desert has definitely been more of our problem child because the rains are monsoonal, and we get a lot of more soil mobility and overland flow in those larger convective events. So what we did is we installed those in September when they get some of those rain events. But we have had problems with trying to stabilize those soils more down in the hot desert. Cold desert has just been generally easier for restoration, and we're just kind of challenged by being down in the warm season dominated precip zones. Yeah.
In thinking about the application of the checkerboard technique, if that ends up being effective treatment, how big are these checkerboards? The little straw squares.
I think you can. Ours are meter squared. Well, actually, the one when our larger plots are 2 by 3 meters. So, but generally, when you install those, they're about a meter squared, and they can be smaller or larger depending probably on the mobility of the substrate you're trying to stabilize. But it's pretty amazing that some of the success they've had in China with reestablishing plants in there, because the seeds, if you seed into there, the seeds stay. They also can capture any windblown seed, and they just stay within there. And what we're seeing is a lot of recruitment, like in the borders of the straw checkerboard. There's a lot more soil moisture and shade in there, so we're seeing a lot of annual plant recruitment in those areas. So we'll be monitoring that, but we don't know if it's annuals that we really want or not at this point.
But so if you were trying to restore a large area, you'd have to have 2 by 3 meter checkerboard, straw checkerboard.
I would probably, if it was highly mobile area, do meter squared, like all connected to each other, yeah. And, well, obviously, I'm the PI, so I don't do any of this work. But the field crews, when they say that they got really good at putting in the straw checkerboards. So you have one person going and laying them out, and then you have another person just coming through with a shovel and pushing them in, and they just pop up like that. And so I think you can get quite fast if you needed to do a larger area. Yeah.
So the idea is that, because it seems like that's kind of a lot of foot traffic to install these checkerboards. So these areas are already so degraded such that
Yeah.
walking all over them.
Yeah, that wouldn't matter. You're working in a degraded, if you went that route, you would be in a highly mobile, degraded soil already. And yeah, whereas you might want to go the polyacrylamide route if you weren't, which is just backpack sprayers, where the glue, the polyacrylamide is diluted in water, and you just spray it on the soil surface. Carrie.
Do people know how stable the community composition is
Over time?
Yeah, like over year to year, 5 years.
That's a really good question. I don't know, like pulling out of my brain, anyone that's really looked at, so especially seasonally, as you ramp up, coming out of winter into spring, who is active in there and what abundance, and if that changes as you go into the heat and drought of the summer. But I think that's pretty interesting to think about. I think some of the cost of doing that and the time has been enough in the past, but now we're getting to a point to be able to answer some of those questions. Be interesting, though. Yeah?
So what are the kind of circumstances that managers are interested in
Yeah, it's one of those things that we haven't had managers come to us yet and say, how do we restore these, as much as we've had a lot of managers say it's not going to work because we've talked to other scientists in the past and it didn't work for them, which is true. But I think there are, especially when there's dust mitigation issues, and if you have, if you work for so you can imagine the Park Service has very different uses, that if a campground went in and they have developed areas that they want to just get back, 10 by 10 meter area of crust near a campground, that would be a very different use than going to BLM lands where you really have highly degraded site, let's say by a water point especially, that you would like to stabilize. Then, they're not banging down our door yet for inoculum and like how to restore these soils, but there is interest in dust, especially, and how to stabilize soils. And so this is kind of like a package of like, well, okay, if you want to stabilize soils and do it with native biotic communities, this is an option. Yeah.
What the crusts that you collect, and if you're saying you could stockpile those, is there any? I mean, that's been a kind of a surface mine reclamation strategy for a long time. Is there, not in terms of the crust, but just for other plants and for the soil itself? But how long does that? I mean, is that good for a year, or is it good for 5 years? Or
That's a really good question, and we don't. So we're in the process. I think ASU is in the process of testing that right now. So. Sergio, for example, we had buckets and buckets of inoculum that we weren't able to use from the greenhouse grown. And so what we've done is we're storing those and going to keep continually testing like chlorophylla content, see if we can, this is a perfect undergrad or master's project, start wetting them up and seeing what their activity is. Do you lose a lot of those organisms? I mean, what we know about these organisms is they can go through a lot, like once they're dry, and if they're stored in the dark and they're not growing anything. So we had some fungal contaminants when they weren't completely dry and had to toss some inoculum, that they should be fine, because that's what they do in the desert. They can go for long periods of time without water. So, but we're talking about like having all those buckets that we have out there tested for chlorophylla and activity. All right, thank you.